Preparation of cell smears. Cell smear is a commonly used technique in which cells are evenly distributed onto a microscope slide to facilitate microscopic observation of cellular morphology, structure, or staining analysis. It is widely applied in hematology, cytology, pathology, and flow cytometry research. Cell smear techniques include push slide method, spreading method, pull slide method, drop dry method, cytospin centrifugation, compression technique. The most frequently used are push slide method, pull slide method, spreading method, push slide method. Prepare 2 clean microscope slides. Use a pipette to place 10 microliters of blood on one end of a slide. Take another slide and touch its edge to the center of the first slide at an angle of approximately 30-45 degrees. Slowly pull the second slide toward the drop of blood to allow the sample to spread evenly at the contact edge. Quickly push the spreader slide in the opposite direction of the blood drop to create the smear. Pull slide method. Prepare 2 clean microscope slides. Use a pipette to place 10 microliters of sample at the center of one slide. Gently place a second slide on top of the first, slightly offset. Quickly and smoothly pull the 2 slides apart horizontally to form the smear. Spreading method. Prepare a clean microscope slide. Use a pipette to apply the sample onto the slide. Spread the sample evenly in a spiral or zigzag pattern to create a uniform smear.